Hello, I'm Won-Suk Kim, BK Assistant Professor in the Department of Electrical and Computer Engineering at Seoul National University. In this presentation, I am going to introduce Nanoelectrokinetic sample-to-answer device that can rapidly and precisely detect bacterial infectious disease. Pathogens can be divided into two categories. One is a virus, the other is a bacterium. Viruses are about 100 nanometer or less. Bacteria are about 1 micrometer. In this study, we used bacteria which are more easily observable. We focused on the development of a device that can diagnose urinary tract infection, which is called UTI. UTI is a representative bacterial infectious disease. Urine contains a certain amount of bacteria, usually less than 1,000 numbers per milliliter. However, when infectious bacteria enter the urethra, the population increases rapidly. If more than 10,000 per ml, you can feel various symptoms. What are worse, complications may occur depending on the underlying disease. Currently, we go to the hospital and get tested when we feel symptoms. First, a multiple-step sample preparation is required to separate only bacteria with high purity from collected urine sample. And it takes more than a day to incubate the bacteria to obtain an observable population. It takes about 3 to 5 days to determine what type of bacteria is infected. So, if the symptoms are severe, the doctor arbitrarily prescribes antibiotics. However, if antibiotics that can kill the bacteria are not prescribed, anti-resistant bacteria develop in our body. Therefore, a device that can rapidly and precisely diagnose UTI is required. In this study, we propose a nanoelectrokinetic micropaper-based analytical pad called Nanoelectrokinetic micropad for quantitatively tracing back the amount of bacteria in a short time. Using this sample-to-answer device, all we need to do is send one drop to the device. This is a scanning electron microscope image of paper. It can be used as a microchannel because it has a pore size of 10 micrometer with the nitrocellulose structure. Paper-based microchannels are easy to parallelize for securing required sample volume for sensitive detection. Using these advantages, several methods of using paper-based devices for concentrating or separating particles have been reported. In addition, various methods for detecting target particles by embedding ELISA or an electrochemical sensors have been reported. In order to develop an early diagnosis chip, it is necessary to manufacture a device capable of both sample preparation and the detection functions. We call this sample-to-answer device. By using a hydrophobic wax, we fabricated paper-based microchannels like this figure. To preconcentrated bacteria, we send nanoporous membrane and electrodes. To detect preconcentrated bacteria, we send nitrosaphine-coated patch. Lastly, to prevent evaporation of the fluid sample, parafilm was coated on the device. Let's see how bacteria are preconcentrated in the device. When we apply electric field across the nanoporous membrane, ion depletion zone is generated from the nanoporous membrane. Then, negatively charged bacteria cannot pass through the ion depletion zone and being to be stacked at the first balance region between the convective drag and the electrophoresis. Let's see why ion depletion zone is generated from the nanopores membrane. A width of nanopores is too narrow, surface charge governs inside the nanopores, so only ions with opposite charges can pass through them. If you apply electric field across the nanopores membrane, concentration distribution is accelerated and going to be non-uniform like this. Here we call ion depletion zone and ion enrichment zone. Complexly, we call this phenomena ion concentration polarization, that is ICP. You can see ion depletion and ion enrichment zone in this movie. This is a conventional concentrator platform using ICP. However, in this study, we utilize simpler concentrator platform for easy multiplexing. When the nanopores membrane is installed at the bottom of the microchannel, a speed difference between the positive and negative ions passing through the nanopores membrane occurs. An ion depletion zone is formed to satisfy charge neutrality, and a preconcentration phenomenon occurs in a microchannel with a sufficiently small thickness. To find maximum and the most stable preconcentrator design, we fabricated three types of the device. First one is just simple microchannel. Second and third one have a converging inlet and a diverging outlet. This is a numerical simulation result of inhibition through the paper channel. In a simple structure, linear decrease in inhibition rate occurs. However, in converging and diverging structures, the inhibition rate converges to a constant value when the fluid reaches the diverging region. As a result, we confirm that the most stable and maximum preconcentration occurs in the 2:1:2 ratio channel design. More than 100 times of preconcentration was achieved at the condition of the 140 voltage. After the preconcentration, the bacteria should be lysed to be detected because anti-drug resistant bacteria have their own enzymes in their cell walls. Therefore, when the bacteria are lysed, a unique enzyme is released, and the amount of the bacteria can be traced back by analyzing the amount of the enzyme. Typical methods for lysing bacteria include thermal lysis, alkaline lysis, and electrical lysis. In this study, we introduced a method for preconcentration and lysis of bacteria on a chip simultaneously. When a negative voltage was applied to the electrode attached to the nanopores membrane, alkalization proceeded so that the preconcentrated bacteria would be lysed, like this figure. The fluorescent intensity of red indicated the population of that bacteria. The intensity increased as a function of time, showing the preconcentration was successfully working. As you can see in this time versus intensity plot, optical detection limit was order of 10 to 4 CFU per mil. To confirm the formation of OH- ions from the cathodic electrode on the nanopores membrane, litmus paper was placed on the top of the device. After applying voltage, the color of litmus paper was gradually changed to indigo color, which means alkalization, and expanded from the nanopores membrane. The higher the voltage was applied, average pH change at the preconcentrated area was faster as a function of time. This plot told us that the voltage more than 140 volts should be applied in order to reach over than pH 10 within 60 seconds for efficient lysis of preconcentrated bacteria. When the preconcentrated bacteria are lysed, the enzymes are released from their broken cell wall, particularly bacteria that are resistant to an ampicillin antibiotic have beta-lactamase enzymes. The beta-lactamase enzyme reacts with nitrosamine, changing its color from orange to red. Early detection of UTI requires detection at a level of about order of 10 to 4 CFU per ml. However, the problem with the existing method is that the amount of bacteria required for color detection is too large. At room temperature, it takes order of 10 to 8 CFU per ml bacteria to detect the color change in nitrosamine and takes about 30 minutes. Under body temperature conditions, order of 10 to 7 CFU per ml of bacteria can be detected after 10 minutes. Therefore, it can be seen that adequate temperature and time conditions are required for rapid enzymatic detection. So, next, we confirm temperature variation at the preconcentration site using thermochromic microcapsules. The preconcentration site should be under 40 Celsius degree to prevent denaturation of protein-based enzymes, and if possible, it is the best to be maintained around 37 Celsius degree for accelerating enzymatic reaction. 4 different thermochromic dyes were installed on the preconcentration site. Each thermochromic dye has its original color, but when temperature rises above threshold temperature, it loses their own color. Let's see these movies. It can be seen that as the voltage was applied higher, many thermochromic dyes lost colors, which means that the temperature rose higher. As described in these snapshots of 140 volt, the temperature can be maintained in the range of 35 to 41 Celsius degrees. This means that the optimum temperature was regulated during concentration over 100 volts while accelerating the enzymatic reaction. Other voltages would give to hot or to cold environments for the reaction. Here are the experimental results. While there was no color change in negative control and order of 10 to 3 CFU per ml bacterial sample, significant color changes were observed in the case of 10 to 4, 10 to 5, 10 to 6 CFU per ml concentration. The detection time would be faster as the inoculated bacterial concentration increased. Through this analysis, we concluded that our proposed platform had a limit of detection of 10 to 4 CFU per ml within 10 minutes. For testing practical utilization, we constructed the cell phone-based colorimetric assay platform. To examine the feasibility of this platform in a clinical practice, human urine samples containing E. coli and Corynebacterium were collected. After 200 seconds of voltage application, the image of detection patch was captured using a cell phone and the red to green ratios were plotted. As you can see in this graph, we successfully enhanced the sensitivity over 1000 times using the device. No color change was detected in the case of Corynebacterium since it didn't have beta-lactamases. In a conclusion, We firstly visualized over than 100 volts preconcentration using nanoelectrokinetic phenomena. Secondly, we proposed an on-chip alkaline lysis of bacteria under spontaneous heating for accelerating enzyme-link reaction. Lastly, we demonstrated susceptible ampicillin-resistant bacterial detection with the limit of detection down to the order of 10 to 4 CFU per mil. We promise that our nanoelectrokinetic micropad would open new era of precision medicine for public health in remote limited settings. Thank you for listening.